This is our Germs Everywhere lab presentation presented by Asia Walter,
Jayla Hannum,
Kendall Jackson,
and Milan Patterson.
Our experiment idea originated from a TikTok where a man tested multiple public bathroom air dryers for how much bacteria they will hold. So as a group, we thought we should do something similar at North Carolina A&T.
The question our experiment was based off was which hold more bacteria, the push button on the paper towel dispenser or an air dryer. We hypothesized that the air dryer would produce more bacteria than the paper towel dispenser push button. We predicted that if we tested the air from the air dryer, it would produce more bacteria than if we swabbed a push button on the paper towel dispenser. We identified the independent variable as the location of our swabs. We took the air dryer sample in Haley Hall and the paper towel sample was taken in Barnes Hall. Our constant variables were the supplies used in the experiment, such as sterile cotton swabs, agar plates, location and time given for the bacteria to grow. And the control group was the E. coli swab and the sterile cotton swab.
For the method of our experiments, for our first experimental group, which was the air dryer, we put a swab under the air dryer till it turned off. And then for our second experimental group, the towel dispenser push button, we swabbed 20 streaks across the dispenser. Then for both of them, we swabbed 3 agar plates and left them to incubate for 1 week. And then at the end, we counted the number of colonies on each plate.
So here we have our air dryer agar plates on the far left, and they averaged about 1.33 colonies. And then on the far right, we have our towel dispenser agar plates, which averaged about 16.67 colonies per plate. And then in the middle, we have our positive control, which had about 50 colonies, and our negative control, which had 0 colonies. So here's our graph that represents our results. The experimental group, which is our air dryers, can be seen here to have a low mean of 1.33. Our experimental group 2 has a mean of 16.67, and this represents our towel dispenser. Our negative control, which had 0 colonies, and our positive control, which had about 50 colonies. So as you can see, the towel dispensers exhibited more bacterial growth than the air dryers. Using about 3 agar plates for each experimental group, we compared the different types, and we observed that more bacterial colonies grew from swabs on the towel dispenser versus the air dryer.
So we can conclude that our results refute our hypothesis that the air dryer would produce more bacterial growth because, like Asia said, there was more bacterial colonies on the paper towel dispenser plates. And yeah.
We felt as if there's a few factors that in the second control group that could have varied our results. When taking the paper towel dispenser sample, we could have swabbed different components of the dispenser because Barnes is a busier public bathroom than Haley, and it could have changed the outcome of our experiment. To improve our experiment, we test various locations to make sure that we get constant results. Taking the results of our experiment into consideration, we'd be more wary of using air hand dryers because it produces more bacteria than a paper towel dispenser. So yeah, if there's unlimited supplies, I'd like to test the sink handles versus the bathroom door handles to see which one accumulates more germs.
So in Germs Everywhere Part 2, we tested the air dryers and paper towel dispensers for bacterial growth and see which one produced more. Because there was a lot of limitations, like the way we tested the air dryer and not having the equal amount of swabs, like we were swabbing different parts of the paper towel dispenser, so the results were not that accurate. So in part 3, we did it again and we avoided these limitations.
For the methods of part 3 and part 4, we had our first experimental group, which is the air dryer again. And then this time we held the agar plate under the air dryer until it turned off. And for the towel dispenser, we swapped 20 streaks across the dispenser. We repeated these for 3 agar plates each, incubated them for 1 week at room temperature, and then at the end, we counted the number of colonies on each plate.
So here are our results for everywhere part 2. And so as you can see, the air dryers had much larger colonies, but still fewer amount of colonies. So they averaged about 5 colonies per plate, while the towel dispenser averaged about 50. They had a lot smaller colonies and a lot more colonies. The positive control again averaged about 50, and the negative control had 0 colonies. So here again, experimental 1 group represents the air dryers, which had about 5 with a standard deviation of 1. And so the experiment group number 2 had about 50 with a standard deviation of 22.91. Negative control had 0, and the positive control had about 100. So you can see the towel dispensers exhibit more bacterial growth again, with the average number of colonies being 50 plus 22.91. That differs from the air dryers, which had an average number of colonies of 5 plus 1. The top or 5 and plus or minus 1. The towel dispensers had a much higher standard deviation at 22.91 compared to the 1 of the air dryers. That means towel dispenser data was a lot more spread out.
So from our results, again, our results refuted our hypothesis. Although the air dryers had bigger colonies and different color bacteria and fungi, the paper towel dispensers, again, did produce more colonies. Yeah, so the only limitation that we weren't unable to fix was using air dryers and paper towel dispensers in different locations. And this would have helped decreasing all the confounding variables, such as, hold on, so sorry. Okay, for our data, we can conclude that the air dryer filters need to be cleaned more often, so not as much bacteria forms. And if this experiment was conducted again, like I said, we'll do it in different locations because Haley is not as used, the Haley air dryer is not as used as the paper towel dispenser in barns. Yeah.
We would like to acknowledge and thank the biology department for providing us with the supplies necessary for completing this experiment, Mr. Brad Robinson for preparing the agar plates used, our TA Chai for helping us with our needs and being helpful resource in the class. Lastly, our peers and classmates.
Thank you.